Welcome everyone to the sixth lecture organized with Janelia and John Curtin School. I'm Steve, and today we're very pleasure to have Tri Phan from Garvin to give a talk on intravital imaging. Tri is a clinical immunologist and a basic scientist with interest in immunology, autoimmunity, and cancer. He heads the Intravital Microscopy and Gene Expression Lab, IMAGE, and the ACRF Insight Center for Intravital Imaging of Cancer Immunity Therapy at the Garvin Institute. And he's quite an interesting fellow where he's attended the medical school at the UCIT and is a practicing clinician. And he then went on to take a PhD at Centenary, developing in vivo mice models to study B cell response to self and foreign antigens. He was then awarded a C.J. Martin and a Keith Murdoch Fellow at the HHMI UCSF campus, where he learned more about two-photon microscopy from Max Klammer and Jason Stader. He's currently a senior NHMRC research fellow, and his work really encompasses clinical work, genomics, CRISPR mouse models, in intravital imaging, and single-cell multi-omics approach, trying to reveal life at the micro scale in a living mice. And today he will be talking about bone, and specifically difficult tissues to image live. So take it away, Tri.
Great. Thanks so much, Steve. And thanks everybody for joining us, whether it be morning or evening where you are. So as Steve says, today I'm going to be focusing on some of our work in bone, but mostly I just want to actually bring out some of the challenges that we faced trying to do intravital imaging in bone, and just highlight some of the things that I think we could have done better, and hopefully, get ideas from the audience in terms of how we can do things better in the future. So before I start, just as a disclaimer, as Steve mentioned, I'm really a biologist. I'm not an engineer, and that means that, to do the sort of things that we want to do, which is to understand, biology, particularly as it might relate to human diseases, we've had to really borrow technologies and ideas from other people. In this context, I guess we borrowed from neuroscience mostly, the use of intravital two-photon microscopy to try to track the behavior of cells in living animals, particularly immune cells that migrate and cross anatomical boundaries quite often in the course of their lifespan. But also, because I'm not an engineer, that also means that quite often we actually have to jerry-rig ad hoc solutions to our and troubleshoot the way we do our intravital experiments. And as a consequence of that, quite often the solutions can be unique to the individual. And one of the things that we're looking towards developing are robust methods that are really independent of the operator, so that anybody can come in off the street and be able to do the experiments. I guess the other disclaimer is that I'm not a computational biologist or a mathematician. And to me, this goes to the heart of the problem of what I think we suffer from with intravital imaging at the moment. That is that quite often there's a built-in bias in terms of what we do. We design experiments because everything's dark, by labeling cells and structures that we're interested in. So that means that we quite often only get to see what we want to see. And so this bias can work very heavily against us. But in addition to that, in the course of an imaging experiment, we generate an enormous amount of data, but because we don't have the language or the tools with which to process that data, we end up binning the bulk of it. The consequence of all this, and one of the things that we want to work towards is that we want to be much more quantitative, because by being more quantitative, then we will improve our precision and also our reproducibility. So the upshot of all this, being I guess, a biologist trying to play in this space, is that we often improvise. We have quite often simple makeshift solutions. So, our experiments may not be optimal. And so one of the things that's really important to us then is that whatever we find, we think it's critical that we then cross-validate that. And quite often that means using orthogonal methods, whether it be flow cytometry or single-cell RNA sequencing. And you'll see what I mean in a second. So I guess my brief today was to take you through our recent paper describing this new phenomenon that we observed, which was osteoclastic recycling. So osteoclasts are these bone resorbing cells. In the course of our intravital imaging experiments in bone, we saw that they were breaking up into smaller cells, which we called osteomorphs. And so really what I want to do today is talk about, the challenges that we faced in terms of doing this study, how we tried to solve them using the sort of ad hoc improvisations that I mentioned. And I really just want to start thinking about whether there were better solutions and better ways that we could have done these experiments. And one of the reasons for this, and obviously, get feedback from you, the audience, but one of the reasons for this is that actually this project took more than 10 years. And really that in the back of my mind makes me think that that's not a very high throughput scenario. So who are we? Well, as Steve mentioned, we're really an immunology lab, and particularly we're a B cell lab. We're most interested in B cells in germinal centers and the memory B cells and plasma cells that generate from them. And we've really, spent the bulk of our time developing ways and methods to image and track B cell fates in lymph nodes. So, this is a very accessible tissue, and it's a soft tissue, and it's easy to penetrate for intravital imaging. And in the course of those experiments, we've developed methods like intravital two-photon photoconversion, where we can actually use the near-infrared laser to switch the fluorescence output from, in this case, a Ki-80 photoconvertible protein from green fluorescence to red fluorescence. So what that means is then, using the laser light, we can switch on cells of interest. And that's very important to us because for the immune system, responses occur over many days, weeks to months, and occur in different anatomical compartments. The response might be generated in the lymph node, but the memory B cells might then leave the lymph node and recirculate through the body. Long-lived plasma cells might then leave the lymph node and go to the bone marrow. So this method then allows us to image and track over large scales and distances. And that's going to be important, I think, for a lot of what we do. So then why would we be interested in bone? Well, actually, bone is actually, as I mentioned, a really important site because that's where the long-lived plasma cells go. And that was originally why we became interested in bone. But in the course of our work, we collaborated with Peter Croucher, who is a bone biologist. And the thing that became really obvious for us was that the endosteal bone surface is a really important niche because this is a site of residence for not only our long-lived plasma cells, but also hematopoietic stem cells. And then in new work, we realized that it was actually the site of residence for dormant cancer cells as well. And within the endosteal bone surface, there are these two types of cells, the osteoclasts, which resorb the bone, and the osteoblasts, which make the bone. And as it turns out that this is a very dynamic process that's continuously happening in our body every minute of every day. So there's this balance between bone resorption and bone formation. And diseases arise when that balance is disrupted. So for example, in osteoporosis, there's far too much bone resorption in excess of bone formation. So this is also really important that because these are where the HSCs reside, this process then dictates what happens during infection, for example, with COVID, during emergency granulopoiesis. It dictates the homeostasis of plasma cells. And also with the cancer cells that spread to the bone, it dictates whether they are active and dormant or become dormant and reside there for a long period of time. So these are the osteoclasts that are resorbing bone that we're really interested in. And one of the biggest challenges, I guess, is that bone, although it's highly dynamic, it's actually really difficult to penetrate because you've got this thick layer of mineralized cortical bone. And what that means is that it's opaque, so you can't get good light penetration. And the bone actually itself has a very high refractive index. And that means that the light that does get through is highly scattered. And so the consequence of all that is that most of what we know about bone, and particularly about osteoclasts, is derived from either destructive static imaging methods like histomorphometry, where the bone tissue is harvested from an animal after it's been sacrificed, or from in vitro cell cultures, where you take precursor cells, which we know are monocytes, macrophages, and then stimulate them with supraphysiological doses of cytokines like GM-CSF and rancor to stimulate their production and activation. So you can get imaging of osteoclasts, but these have largely been in vitro. Now, the other thing I should mention about osteoclasts is that they're multinucleated, and at least on these static images, they're these big round cells of about 150 to 200 microns in diameter. So they're really large cells. So what we have learned from these static and in vitro methods is basically summarized here, that osteoclasts are formed from the fusion of multiple CSF1 receptor positive precursors. And so this is why they're multinucleated, and that multinucleation is actually really important in their bone resorption capabilities. And one of the big drivers of that multinucleation is a TNF superfamily member called rancor. And this is actually the master regulator of osteoclast differentiation. So monocyte macrophage lineage cells, when stimulated in the presence of rancor, will fuse and will form osteoclasts and resorb bone. But one of the other things that we learned from these in vitro studies is that actually what happens is that the osteoclasts are short-lived, and at the end of their life cycle, their very brief lifespan, they undergo apoptosis and die. So we wondered whether this is in fact what actually happens in vivo. So the traditional approach to intravital microscopy, in fact developed at Harvard by Steve's old mentor, Charles Lin, used the idea, bypassed the thick cortical skull by riffing on the idea that actually in the mouse, the skull plates are not fused. And so you have these sutures. So, you know, newborn babies, the skull plates aren't fused, but as the human skull develops, those sutures fuse. But in the mouse, it doesn't fuse. And what that means is that it leaves this small optical window through which you can penetrate. And so in a typical imaging setup shown here, the mouse is immobilized and then imaged through the skull through this bregma, which is the confluence of the sutures. And so Christina Low-Kelso and David Scatterton, using this method quite famously back in 2009, were able to image the hematopoietic stem cell niche in the skull bones. But the problem, of course, is that the skull is non-weight bearing. It's derived from intramembranous ossification. And the upshot of which is actually, it's not the site of most human disease, which is actually in the long bone. So one of the other ways to image, so that method actually is relatively minimally invasive. It just involves a suture, an incision in the skin to expose that the sutures underneath. But other people develop far more invasive approaches. So there's two examples shown here. In this example, to overcome the problems of the thick cortical bone, the investigators use an electrical drill to shave bone away. And then in this example here, developed specifically for long-term longitudinal imaging, then use a surgical implant, essentially tubing that will allow you to use a microendoscope with a green lens to then image the bone marrow space. But the problem with invasive approaches, particularly in the bone, is that bone is actually, as I mentioned, exquisitely sensitive to pressure, to loads. You know, as I said, it's constantly being remodeled. And so any perturbations to that bone is going to change the biology underneath. And so we really need a minimally invasive approach, because otherwise these sorts of imaging methods will change the biology that we're looking for and will therefore generate an artifact. So we tried to develop a different approach, and it came from the realization that actually if you just make an incision in the skin overlying the tibia, your shin bone, you can see actually here the red marrow space underneath. And that was telling us that actually the bone, at least in the tibia there, was thin enough for light to penetrate through. And so if you just remove the overlying muscle and the fascia, then you may be able to penetrate it with a near-infrared laser. So this is our imaging method, sort of outlined here in a schematic form. So essentially, this is the mouse anesthetized, the skin is shaved, an incision is made, the tibia is exposed, and then the leg is immobilized. And one of the things that we need to do is we use a silicone grease to create, and then thermal conductive tea putty to create this imaging chamber that needs to be waterproof, because we image directly without a cover slip. And I'll tell you why in a second, hopefully. But the key thing here now is that this method allows us to image a weight-bearing long bone, which is derived from endochondral ossification. And so this is where most pathology occurs. So, oh yeah, and the other point, I guess, is that because it's just a simple incision, at the end of the imaging session, we just suture up the skin, and we've been able to do longitudinal imaging over months with this method. So one of the problems, though, with this method is that because we want to see subcellular resolution, we're using a high magnification, high numerical aperture lens, which has a short working distance. And so, and obviously the lens is actually quite bulky itself. So that means that we don't use a cover slip, so that we can get as close as we can to the tissue. And the other problem we have is motion artifacts from breathing, but also from transmitted arterial pulsations. So I'm just showing you here the ideal situation. So this is the tea putty in overview, and this is meant to represent the tibia, and this is a meniscus. So ideally, you want a nice, small, stable meniscus. Sometimes we have problems where the tibia is at an angle, so you can only image a very small part, and it's not a flat field of view. Other times we have a very large meniscus, so we're losing a lot of the light. And then the common problems are, because it's an immersion, you get evaporation of the meniscus, or worse still, you haven't achieved a waterproof chamber and there's a leak. And we try to overcome that when using immersion liquid like Immersol. And then obviously this problem where we failed to isolate the hindquarter, and so there's transmitted breathing or pulsations, or because the mouse is unstable because the anesthesia is not deep enough. Okay, so, and then this is how we typically image. We would take Z-stacks, are typically 3 micron steps, although, for fine imaging, we may take 1.5, but not much smaller because of the point spread in the axial plane. And we normally image at 30-second intervals. So we capture these Z-stacks, typically 30 Z-stacks, 30 seconds apart. And so we actually end up with a not unreasonable imaging volume of 425 by 425 by 150 microns. But of course, that's actually a very small part of the tibia bone. And then because of the limitations of anesthesia and the fact that our animal ethics requires us to check on the mice very periodically during the imaging session, we're limited to only 30 to 90-minute imaging windows. So if there's biology happening over much longer periods of time, which is what happens in the bone, we then actually have to interrupt the imaging and stitch the Z-stacks together if we want to capture the whole tibia, or stitch time together if we want to capture a big, like a 7-hour movie. So of course, this creates problems for us because in terms of space, we quite often want to see at least subcellular resolution. We want to see fine cellular processes. We want to see maybe even organelles. But at the same time, we want to see, capture larger volumes because particularly for the immune imaging work, our cells migrate and are very motile, so they may leave the imaging volume if it's such a small imaging volume. And then also we operate on different scales in terms of time as well, because the biological processes may be occurring very rapidly on the one hand, so you want to be able to capture much higher temporal resolution in 30 seconds. But then on the other hand, because the overall process may take hours, then you also want to be able to capture that longer time window as well. So this is actually a common problem for us, which is a problem of scale. So the other problem, of course, is imaging depth. And as I mentioned, the cortical bone is quite thick. And as it happens that in the tibia, we usually gravitate towards a small area where the bone is naturally at its thinnest. And an example here, I think it's only 170 micron thick. And of course, we have a very old setup with a laser that can only get up to 1060 nanometers. So we typically image at 920 nanometers. And what that means is that, as you can see here, we can only penetrate typically about 100 to 200 microns below the cortical surface. And it may well be that with other lasers and longer wavelengths will penetrate deeper. But for us, at least luckily, a lot of the biology that we're interested in, like the osteoclasts, they reside on this bone surface here. The dormant cancer cells that we're interested in reside on this dormant surface here, and the long-lived plasma cells, we think, also reside in the same place. So that's great for now, but really we are missing a lot of biology. So this dark space here, and this is in the bone marrow where also there are niches for other stem cells, cancer cells, and obviously a lot of blood cells that may be really important. So that brings up this problem that we have, is that actually we may well be missing a lot of really interesting biology with this method. Okay, so what does it look like? So this is the first iteration. So this is from 6 years ago now, where Michelle McDonald took on this rather heroic task of trying to capture a cancer cell, in this case, a multiple myeloma cell, which is labeled green, arriving in the bone and then colonizing this endosteal bone surface. And just so you know, like this one, brief few seconds of image of a movie that I'm showing you, that actually took us something like 50 hours of live animal imaging to capture, which brings up this problem that we have, is that a lot of the time we're trying to image very, very rare cells doing very, very rare things. So this really is like the ultimate low throughput experiment. And I guess the analogy I like to think of is that it's kind of like, one of those David Attenborough wildlife documentaries where some poor cameraman's been stuck in a bird hide for 6 months waiting for, like a blue-footed booby to do its mating dance that it only does for like 5 minutes once a year. So we really need to think about ways that we can do this better, if only for the sanity of the people doing the experiments. Okay, so we want to look at osteoclasts, and osteoclasts are formed from the fusion of macrophages. And what we really need now is a way to light up the cells for imaging. And so we took advantage of that fact that they generated from fusion of macrophages by thinking that if we had a red and a green macrophage, when they fuse, you'd generate a cell that was multinucleated, but also both red and green. And so the fluorescent protein we used, the choice anyway, we were kind of limited. We were kind of like stuck with TD tomato and enhanced GFP. And of course, as you well know, there is a very big difference in the brightness of TD tomato compared to EGFP. And once you go through thick cortical tissue like bone, those differences become even more marked. And so we will see hopefully what I mean in a second. But anyway, so to do this, we have 2 donor mice, one in which the macrophages are red, one in which the macrophages are green. We harvest the bone marrow, mix it, and then use it to reconstitute the bone marrow compartment in a lethally irradiated mouse. So then this mouse now has bone marrow that can generate both red and green macrophages. And so the osteoclast, stochastically that would be generated would be from the fusion of a certain number of red and a certain number of green macrophages. And so hopefully we'll see cells that are both red and green. Okay, and one of the great things we think about this and why we came up with it was that we had tried imaging osteoclast reporter mice, but unfortunately they were quite leaky and we couldn't be sure that we were looking at osteoclasts. Okay, so this is what actually an osteoclast looks like in vivo in a live animal. So very different from that static image I showed you earlier where the cell was this big rounded cell sitting by itself. So in actual fact, it's a big cell, but it's got all these dendritic processes and these processes are contacting neighboring cells. So it's forming an interconnected network all over the endosteal bone surface, raising the possibility that actually there may be some sort of cell-to-cell communication that allows them to coordinate the activities in terms of bone resorption, and for other cells to regulate their activity and couple that to bone formation. Now, one of the things I guess to point out is that you may notice that the red signal doesn't actually co-register as well as or completely with the green signal, and this is the problem that I alluded to earlier, that it may well be that due to differences between wavelength and point spread function, but essentially the green signal is not as strong as the red signal, and obviously the red signal is quite saturating in imaging, and there's a reason for that, one of which is that we wanted to capture these cellular processes and the connections and interactions with the nearby cells. Okay, so already in the steady state, they look really different. We know that rankle stimulates osteoclastic bone resorption. So what happens if you inject rankle? And so you can see here in this example where we think what's happening is that these and so some of these imaging, just to make it clearer, we've just removed the green channel. You can see actually that not only do the cells, pull back their processes, but they actually start moving together, and then they fuse to then form an even bigger activated osteoclast. And then we're just sort of like trying to highlight this here by this cell fate mapping that we do, which I'll tell you about in a second. But we can see that they fused when you examine them through the different Z stacks, and also by examining in 3 dimensions to see what's going on, and then also by looking at the sharing, particularly in this example of the green fluorescent protein, because one of the cells doesn't have much green fluorescent protein, this one down here at the start, but after fusion, you'll see that there's green fluorescent protein in the cell. Okay, so that's kind of like not unexpected, because we know rankle stimulates osteoclastic bone resorption. So what happens next? Well, this was kind of like the surprising part, and what we didn't really expect. And what was happening was that if you actually watch the cells for longer and longer period of times, in this case 3.5 hours, you'll see that actually the cells start to break up, but they don't break up into small subcellular fragments. They actually break up into large cells, and these cells are actually still quite motile, and they're still migrating around. So we saw rankle-induced fusion before, and now we're seeing in vivo for the first time rankle-induced fissioning of the cells. Now, of course, I mentioned to you before, the dogma from the in vitro studies was that osteoclasts undergo apoptotic cell death. And so the question arose was actually, is what we're seeing the fission of the osteoclast into smaller cells, or the breakup of the osteoclast into small apoptotic subcellular fragments? And certainly, although the dogma says that the cells are short-lived and undergo apoptosis, as recently as 2 years ago, really beautiful work from Frederick Geismann's lab using parabiosis showed quite clearly that osteoclasts can be much longer lived than the dogma was suggesting. And so, I guess the wheels were already starting to come apart, this idea that the cells undergo apoptosis. So we looked and looked, and in fact, in well over 100 hours of imaging, we really could not see any apoptotic events. And so we kind of like decided, okay, we need to actually induce apoptosis to see what it looks like and compare it to what we were seeing with the fission. And the way we induced apoptosis was to use the near-infrared laser to actually irradiate the cells and essentially give it a burst of lethal irradiation. And actually, when you do that, hopefully the movie will loop, you'll see that now it's completely different. We've kept the green channel on this time just to highlight these red macrophages that have actually come into the picture. So, for example, if you just focus on this cell here, as it starts to undergo apoptosis and die, it starts breaking up into smaller fragments. These macrophages come in and start interacting with it. And the key difference is that the fluorescence completely disappears over time, as presumably the fragments are ingested and degraded by these macrophages. So we tried our best to be as quantitative as we can in terms of describing this phenomena. So for example, in the cell fission, if we use these sort of cell fate maps or what we call underground maps, because they look like the underground train station network in the UK. So here's your osteoclast undergoing cell fission into daughter cells. And each time it fissions, you still have a cell body. And even though the volume has increased, there is still volume at the end of the fission process. And this is what it looks like in terms of interactions with macrophages. The difference with apoptosis is that after the laser photoablation, each time the cell breaks up, it gets progressively smaller and smaller until finally at the end of the imaging, you see that there's actually no volume left. And there's a really big influx of macrophages coming in and interrogating the cell, the fragments, and then taking it up and degrading it. Okay, so if that's fission, then why is, what's happening to these daughter cells? So this time now, Michelle imaged for even longer, 7 hours. And in this movie, not only do you see the breakup of the cells, and you'll see that as the cells fission, you get these sort of like nanotubes that form as they pull away from each other. So these are retraction nanotubes, so you can see one there. So as the cells fission and recoil from each other, the other thing that happens is that some of them starts migrating towards each other as well, and then you end up with them fusing again. So now we're seeing a cycle, and so we've tracked it here with this map, and you can see now why we think we called it the underground map. Is that cells migrating together and fusing, and also cells fissioning and then fusing with other cells. And so we see now that there's this cycle where an osteoclast will break up and then reform again, and that kind of reminded us a lot of the Mighty Morphin Power Rangers. And just so to have a bit of fun, because these were red and green Power Rangers, we decided to call these cells osteomorphs. But are these cells for real? And I think, like I mentioned at the beginning, for us, cross-validation is really, really important. And so I'll just briefly, okay, so I mentioned that before. So I'll just briefly go through the next steps. So, okay, so this is what we think is happening. So instead of just undergoing apoptosis, there's now an additional cell fate, which is this fissioning into osteomorphs, and then these osteomorphs then recycle back and form osteoclasts again. And of course, how do we cross-validate that? Because they're fluorescent, and because we're immunologists, we can do flow cytometry. And this is showing that actually the double positive cells, so the double labeling that we use, are the cells that actually have accumulated this fluorescent bisphosphonate, zoledronic acids, indicating that they've derived from the osteoclast that's resorbed bone. And also because we can do flow cytometry, that means we can sort them. So we sort them, we can show that they can form osteoclasts, at least on Calvaria, in ex vivo cultures, but also more importantly, it means that we can do single cell RNA sequencing to look at the transcriptional state. And if they were a separate cell state, then that should be quite clear from that analysis. And this is just very briefly showing you that when we sort cells from the bone marrow cells from the bone marrow in black, and from the bone, and we sorted single positive cells, so green only, red only, and then also triple positive cells, green and red, that have accumulated bisphosphonate from these two tissues, and done dimensionality reduction, 3 populations really stand out. So you have these cells coming from this population here, which are largely made up of cells coming from the marrow, but also from the bone. And these are largely either red or green cells. So these are the single positive cells. So we think these are the monocyte macrophage precursors that then go on to form the osteoclasts. On the endosteal bone surface, so mostly the gray here, are these triple positive cells. So we think that these are largely osteoclasts. And then on in the marrow space, mostly the black here, you also have triple positive cells, which we think are the osteomorphs. And you can see when you visualize it, it's quite clear that there's 3 distinct populations of cells. So 3 distinct populations indicating that the osteomorphs are quite transcriptionally distinct from the osteoclasts. And you can see here that actually, even though they're distinct, they do share a lot of their upregulated genes with the osteoclasts, particularly the expression of a number of these canonical osteoclast genes. That was just some cockatoos for those who aren't familiar with Australian bird life. And then just very quickly, we showed that these genes are actually very important and functional, because when they're knocked out in this OBCD program, you get bone phenotypes. So either the bone's shorter or longer, thicker or thinner, and also importantly, functional bone effects, either the weaker or stronger. And one of the things I want to highlight was that 2 of these genes, BPGM and FBX07, are were unique to the osteomorphs and weren't upregulated in the osteoclast, suggesting that actually this cell state not only is distinct and a separate cell state, it can actually be a very important source of gene discovery in terms of what's important in osteoclast, osteomorph, and bone biology, and hopefully also then in terms of understanding diseases. And so when we look at human diseases, in this case, the human biobank, where they've measured estimated bone mineral density as kind of like a surrogate mark of whether patients have osteoporosis or not, a lot of our osteomorph genes are actually enriched in genes that are associated with estimated bone mineral density, indicating that actually they're important in human diseases as well. Okay, so really what's next for us and what we do? I guess for us, the big question is, you know, why do these osteoclasts recycle? Why do they break up into these osteomorphs? And how can we answer this, you know, moving forward? Having, as I mentioned to you, done things really badly and taken this really long road to get to where we are there easier and better ways to get to this next question, which is, what's the reason for the osteoclast fission? So this is kind of like one of supplementary figures from the paper. And I guess one of the reasons why I wanted to bring it up was just to show you that actually, if you added all this up, then really this is, you know, 87 hours and 46 minutes of imaging. Now, of course, that's what's gone into the paper, but, you know, at least twice as much time has gone into imaging experiments that have just, you know, not worked, whether, you know, the problems that I mentioned before, you know, problems with the meniscus, problems with, you know, flatness of field of view, problems with the surgery, etc. But I guess the other thing I wanted to bring this up was to highlight that actually, you know, these fate maps that I mentioned before, you know, our very complicated, sophisticated fate mapping exercise was an intern sitting there and watching each movie frame by frame, Z-stack by Z-stack. And so for 87 hours, 46 minutes of imaging, you can at least multiply it by 10 times in terms of the amount of time that's taken to analyze that data. And so, you know, in the back of my mind, I guess, there must be a better way for us to do this. Okay, so I told you before, this is what we did wrong. I guess the problem is that, you know, we're biologists, we sort of like don't have this background expertise. And so moving forward, I guess, what have we learned and what do we think we can do right or better next time? Well, I guess one of the things is to realize that actually what we needed is an interdisciplinary approach. So we really need a situation where, you know, the biology talks to the, you know, the engineering side and talks to the mathematics side, so that, you know, the pre-analytical, analytical, and post-analytical phases all come in together. And so what that will allow us to do is develop custom technologies that are fit for purpose. And I'm going to show you something that Steve's done for us in a second that speaks to that. And really, this is about engineering solutions so that we can be far more reproducible in what we do. And then also looking beyond that with, you know, into the future, taking advantage of technologies that really would allow us, for example, to get deeper, you know, for example, using 3-photon microscopy, and then ultimately maybe to do label-free imaging, for example, in patients themselves, not just in animal models. You know, on the computational side, I think, we really need to get more involved with people who can provide skills and capabilities for us to take more agnostic approaches that can use all the data, not just, be blinkered in looking at the things that we're looking for. And that might incorporate not just the imaging data, but all the metadata and also additional data surrounding it. So you have this sort of like multimodal integrated big data approach. And what we're hoping that would do then is allow us to be far more quantitative and far more predictive and take advantage of, things like, agent-based modeling or emergent behavior or whatever your artificial intelligence method of choice is. And I think the upshot of all of that is that then we'll be able to have, better animal models to allow us to do much more optimal experiments. The sort of experiments that we really want to do are those that span those scales, that problem of scale that I mentioned. We want to be able to image molecule to cell to organ, and we want to image, in the maybe even the milliseconds, but then, also, as you saw, over hours and then days and possibly even longer. And then the imaging for us has largely been really descriptive. And I think that analogy with, David Attenborough wildlife documentary is not a bad one, because mostly it's about seeing things. And for us, actually, where we want to take our intravital imaging is to a place where actually we're not just seeing but we're manipulating. And that manipulation allows us to get to mechanisms. So, doing things like 2-photon caging and then niche-seq. So I'm going to sort of wrap it up here and talk about briefly a couple of slides about this very exciting new initiative that we're involved with Steve, and that's the ACR for Insight Center. And this whole program is about trying to look into the dark spaces in tumors. So that's our enemy, really, the dark spaces. And so here is the endosteal bone surface. This is the marrow space that we're not very good at looking inside. And here is a single solitary dormant cancer cell. So the tumor cells are green, and red is a label, a dye that gets diluted when cells divide. So the dormant cancer cells, because they haven't divided, have retained the dye. And here's a growing tumor cell. And the main points I want to make with this slide is, one, the dormant cell is sitting very close to a growing tumor cell, suggesting that the niche in which it resides is actually a very small, localized in space and obviously in time as well. But we don't know what's in the black here. And we don't know what makes up this dormant cancer cell niche. So not only do we not know what's in there, we don't know what to label to look in there. And so, of course, we can take a candidate approach, label stromal cells, etc. But really, to be more agnostic and robust and not to miss anything, we need something different. And so one approach might be to photoconvert using that method I mentioned at the beginning, this region, and then isolate the cells, and then do single cell RNA sequencing, for example, and then annotate the cells that way. So that's niche-seq. So Steve has developed this brilliant technology, which hopefully he will implement for us and allow us to do imaging at much faster speed, which is really critical, but also allow us to image bigger volumes. And using the adaptive optics technology, the imaging quality should significantly improve. And that will overcome a lot of the problems, I hope, that we're having trying to penetrate that deep cortical bone tissue that I mentioned at the beginning. And then also Paul Timson, who I think will be talking later on, and we'll probably talk more about this technology that they developed of Gaylean, which is at the other end, post-analytical, which and can also be implemented and used on the fly. But basically, it's a method for stabilizing imaging. And so normally data that you would bin and not be able to use, you can then still recover and retrieve lost data from motion artifacts. So I think that's it. Really, hopefully given you enough for you to think about how you can help us improve our bone imaging. And definitely, if you have any suggestions, please drop me a line. And I'm just going to finish by thanking Peter Croucher. This has been an enormously exciting adventure with him over many, many years, trying to develop these methods for imaging bone and studying what I think is really the final frontier in terms of immune imaging anyway. Michelle McDonald, who valiantly did all that imaging, and then Wang Hua, who's been instrumental in confirming the existence of these cells through those orthogonal approaches, and our collaborators, and obviously our funding. And then I'm going to stop there, and hopefully you'll have suggestions or questions for me.
Fantastic, Tri. Wonderful talk. I have a few questions. Probably start off with 3 questions. The first one, you talk about weight bearing bone. Right. And I guess it's not very evident or clear to me weight bearing and the importance of weight bearing. I guess gravity and some sort of compression, mechanical. Is that what you're?
Yeah, absolutely. Yeah. And so it's really interesting because embedded within the bone is this network of cells, terminally differentiated cells called osteocytes, and these osteocytes send their tiny process, and which we can image, and they all communicate with each other. And it's thought that they have pressure sensors. So I think things like piezo-1, mechano transducers are really important in the functioning of these cells. So they sense mechanical load on the bone and then use that signal to then communicate with the osteoclast and the osteoblasts to coordinate bone resorption and bone remodeling. And of course, as you know, one of the big problems with space travel is that in the astronauts coming back, they have terrible bone loss because that whole process gets uncoupled. And in fact, there's a few experiments that we want to do, and I quite flippantly think that actually we should try and load it onto the next Virgin Atlantic or whatever SpaceX satellite. So yeah, bone is really interesting because gravity is really important, and actually that communication might extend beyond the bone as well, in terms of the muscle tissue, and then the bone is actually an endocrine organ. It's produces and secretes a lot of hormones, so there's a lot of action at a distance as well.
I see. And I guess the next question, going back to the microenvironment, is that there will be some fluid shear, might it be quite low, and when some of the videos when you show that the osteoclast was reaching out, would that be because it's sensing some fluidic imbalance within the microenvironment?
So we don't really know, to be honest with you, but I think it's really interesting because you're absolutely right, because in the steady state, even though not much is happening compared to say when we stimulate with rancor, there is still a lot of activity going on. Yes. And there's, as you say, there's a lot of reaching out between one cell and the other, and there's a lot of contact going on. So what's actually happening is happening probably at a scale, and that's one of the reasons why I mentioned that for us, one of the problems with scale is that we'd want to be able to image the cell, but we also want to image the, hang on a second, this is just my dad who's in hospital, just give me a second. Hello, Bob.
So Steve, we need a plan to image the dark space.
Yeah. Absolutely.
Sorry, Tri, just jumping in while you are on the phone.
Hi Liz, how you going?
Good.
So just going back to fluid, I guess the last question I have, apart from the complexity of the microenvironment, is that there's an image you showed multicellular to single cellular. Are you sure it's single cellular?
No, we're not.
Osteomorph? Because it could be very different, right? I mean, that transition.
Yeah, no, absolutely. So actually, we think completely conjecture, speculative. We think that that the cells are breaking up for a reason. We want to drive at that reason. We have some ideas, but they're really thought bubbles at the moment. So I don't want to mislead anybody. But one of the things is that some of these osteoclasts have a dozen or more nuclei, and when they break up, you would imagine that as they break up and partition, some of the daughter cells will have 2, 3, maybe even more nuclei as well. And so I think what we're capturing there is still something that's quite dynamic, and that's still in a state of flux. So whether they divide even, fission even further, until they become mononuclear, or whether that initial 1 or 2 fission processes is enough to, you know, for whatever reason that they fission in, you know, whatever triggered it in the first place. I mean, but definitely we have looked, for example, using nuclear dyes, also by flow cytometry, for example, at the cells in the marrow that we think are the osteomorphs. And when we do that, you know, yes, a lot of them look like they have 1 complement of DNA, but some of them look like they might have 2 complements. So yeah, there's definite possibility that actually a lot of them are, you know, oligonuclear, if not mononuclear. Cool.
Yeah. I guess the audience, any questions?
I have several questions. So I'm interested. So first, thank you for the inspiring talk. So as a people from engineering field, it sounds like we have a lot of work can do. We can do some contribution. So I'm curious. So what type of objective did you use for the image? So the reason I ask this question is because I guess normally we use water dipping objective. So but for the bone image, as you mentioned, so the reflective index is quite high. So the water dipping objective may not be suitable for you.
Absolutely, yeah. So I think this is one of those examples I mentioned where I think, you know, we kind of do what we know how to do, and so it is it's your standard 1.0 NA 20 times water dipping objective. And you're right. I mean, with a refractive index of 1.55 to 1.64, and water being 1.33, that's a really big refractive index mismatch. And so we have a couple of years ago, I can't remember what happened with it, but we did try to look at materials or that might actually be closer to the bone in terms of refractive index. And I guess one of the other things I forgot to mention is that to try and overcome the big bulky objective, you know, one of the things that we actually want to try and use, and Steve hopefully will build one for us soon, is a micro endoscope, you know, that's a bit narrower, that will allow us to get closer to the tissues. But yeah, no, so you're right, we're using the wrong immersion liquid.
At the end of your presentation, so you show the adaptive optics helps improve the image quality a lot.
Well, that's Steve, okay.
Yeah, that's very exciting. So yeah, so I was wondering, so before and compare before and after the adaptive optics, what's quantitatively the spatial resolution? So I want to see how much improvement. Yeah, I'm interested in that.
So hopefully our new microscope with the adaptive optics will be online in a few months, and I'll send you the first experiment.
Oh, I see. So you haven't tried yet. So
we haven't tried yet. No, it's all kind of like what we're hoping to do, kind of like the sort of solutions that we're hoping might help us.
I see. I see. Yeah, I imagine that will be very helpful to address the reflective index mismatch. Cost aberration. So Steve, what type of adaptive optics method did you use?
We've done adaptive optics rastering. So instead of looking at the full field, we partition the segments to correct, and instead of correcting individual areas, we correct for every single lines in a polygon scanning unit. And what we found was that if you were to partition just in lines, you can get up to recover 80% of what's lost. So that was very helpful for fast acquisition and fast correction.
Cool. Did you use direct wavefront sensing?
No, indirect. It's indirect wavefront sensing.
Right. That. Yeah, I'm also thinking, because Tri mentioned the co-registration issue of 2 colors.
Yes.
So imagine if your system built with aberration, so then of course different color will experience different aberration. That's may cause the issue contribute to the issue you saw. Yeah.
Yeah, absolutely correct.
Yeah, I think you can get much better image and address this issue.
Yeah, and then just on top of that, like we've been playing, at least in the lab, with a lot of cylindrical capillary tubes to try and mimic all those very large curvatures, and we have been able to correct for up to 80%, recover a lot. So using that model to try and predict when we get to the experiment in vivo, what sort of aberration we'll get.
Cool.
Yeah. And you're right, Chen, because when we have tried to measure the chromatic aberration in our lymph node, we get a little bit, not too much, but in the bone, everything is just magnified,
right?
Because of that cortical bone that you have to go through.
Cool.
My last question may be very naive. So you mentioned, the dye, you use the dye to label the different type of cells. So I mean, since you are doing the in vivo image, right? Would that be toxic to the sample? And in the future, if you want to doing the test on human, I don't know if that's their plan. So
yeah, with the humans, yeah, you're right. I mean, with the humans, we're looking at label-free imaging, so no dyes, and then so most of our imaging is done using genetically encoded fluorescent proteins. But for the dormant cancer cells, we do use a dye label because it's a syngeneic cell line that we then inject into the mouse. And that dye, you're right, like all dyes, do have a certain degree of toxicity. But I guess we're hoping that the cells that do make it in and get to the niche and engraft have kind of like overcome that. They've sort of like been selected as the ones that have survived the toxic effects from the dye labeling.
Cool. So so you talk about the label free. So
yeah.
So that's, yeah, so, right. So would you consider other like modality for imaging or even consider spectrum?
Oh, yeah, absolutely. I think, I guess, well, we've mostly used optical imaging, particularly 2-photon. I mean, about, oh God, it's about 10 years ago now, we developed a microendoscope that sort of goes on to the end of a colonoscope for imaging in patients with inflammatory bowel disease. Our original idea working with the company was to actually develop a 2-photon solution, but we came into a lot of regulatory problems in terms of laser safety, etc. So we ended up just using a confocal system, and that was sort of like, really suboptimal, because there it was just really contrast, autofluorescent contrast imaging. But definitely, I think there are not just optical but non-optical solutions for label-free imaging. A lot of which, but also, a lot of the other modalities, I think, have really significantly improved in terms of their resolution. And so, I'm sort of like picturing a time when we'd be able to do multimodal imaging that captures a lot of these different methods in the same sample. Whether it be, using Raman scattering or whatever. I mean, I think that's kind of like where I sort of like step back and sort of look to people like you guys and say, well, what's possible, what's available, and then try to make the most of that.
Cool, thank you very much. Very exciting.
Right.
I've just lost Liz. She actually put her hand up, but she had to go off. I guess we maybe a little bit that we're looking at megakaryocytes in bone, actually. And one of the things that Liz is extremely interested in. Have you guys looked at that yourself?
Yes, so megakaryocytes are kind of really interesting because, Uli von Andrian about 10 years ago, using that method for skull imaging, imaged these megakaryocytes sitting next to a blood vessel and pinching off the platelets. Yep. Now that was in the skull, and I'd be really curious to see how they look in the bone marrow space. And so definitely, I think, you know, like I said, that dark space is going to be really interesting, because from the hematology point of view, from the hematopoiesis point of view, I think there's a lot of really exciting things that happen there. But also, I think it's far more vascularized. The blood flow is very different from the flow on the endosteal surface. So I can imagining that actually, for example, the cancer cells or the plasma cells that do get to that endosteal surface, they first need to colonize the marrow space and then make their way through the vascular channels, through the sinusoidal network to the endosteal surface. So that's what we're missing, I think. And we really need to capture. Yeah, the deeper we get, the more interesting the biology, I think, will uncover.
Yep, cool. Any more questions? I guess there was a few people that have to run off. I personally enjoyed it, and I think there's going to be a lot more to do at the Insight Center, and something that I really would enjoy looking at, solving the refractive index with the right objectives and the right immersion medium. And I guess any last questions?
I've got a question.
Right.
Hi, Tri. Thanks for your talks. Very interesting. So I was wondering, is the genetic expression, have you been able to determine if the degree of fission, the number of fission events, is it discrete, where you see one fission already triggers change in gene expression?
Yeah, look, that's a really interesting question, and one that I just don't think we have the right resolution and the right method to track at the moment, because I do think that probably these cells are in a state of flux, and I think what we've captured, at least in terms of the transcriptional state of the cells that were in the marrow, may be a slightly more stable version to what we're seeing in the movies, where the cells are fissioned, and I think they're still active, and they might just fuse again. Definitely, I think a lot of the transcriptional changes that we think are a cellular stress response, and you can imagine that osteoclastic bone resorption is an incredibly stressful process. It's highly energetic. You've got a cell that's formed a sealing zone and then is actively pumping acid into that environment with lots of proteases and degradative enzymes. So it's generating a lot of free radicals, etc. And I think there's also a potential for gene toxicity, genotoxic effects as well. So I just think in that environment, there may be a lot of reasons for the cell to then want to fission, and what that fission might allow it to achieve. And so I'm kind of like imagining that it may well be that the fission products, the daughter cells, may be actually quite heterogeneous, not as homogeneous as that population that we saw quite tightly clustered together. And that heterogeneity might reflect some of the steps that you are, I think, are alluding to in terms of their gene expression.
Right. And I'm guessing these fission events, do you think that they relate, the sites where the fission occurs relate to the porosity of the bone, where the fission occurs at regions of lower porosity, where the spacing is much lower?
Yeah, that's a really interesting question. So we want to get to a lot of these things, I think, and a lot of the key to that is actually going to be able to work out how to measure or mark those indicators that you're alluding to that might be driving the fission. And so that's actually, an area that we have someone now who's just started, who's sitting down and thinking very hard about how to go about, looking at exactly those sorts of questions that you're asking.
Cool.
Does anyone have any questions? If not, we could end off the session. Thanks, Tri, again for the talk. Thanks everyone for the questions. It's been a joy, and yep. This session will be uploaded on YouTube, so I'll spend more time
Okay,
editing.
all right. Thanks so much, everyone. Thank you.
So, thank
Thank
you.
you.
Thanks, Tri.